In this longer video, we're going to explain antibody structure. So this is the general structure of an antibody. Now, obviously, we should remember that antibodies are made by plasma cells, which are a type of B lymphocyte. And their role is to produce and release specific antibodies that have complementary antigen binding sites for a particular foreign antigen. Now, this is a protein. It's made from 4 polypeptide chains. So 4 chains of amino acids linked together with peptide bonds. We describe it as having a quaternary structure because any protein that consists of more than 1 polypeptide chain has a quaternary structure. So we've got 1 chain here, 1 here, 1 here, and 1 here. In the center of the Y-shaped antibody, you've got 2 longer polypeptide chains, and these are called the heavy chains. So there's 2 of those longer polypeptides. And then on the outside of the Y-shaped antibody, you've got 2 shorter polypeptide chains, which are called the light chains. Now you can also see that these 4 polypeptide chains are held together with bonds, which I've drawn here. These bonds are disulfide bonds, so strong covalent bonds, holding that quaternary structure together. What else do we need to know about the structure of an antibody? Well, if I just bracket kind of this part here, so this section of the antibody, this is known as the constant region. And it's known as the constant region because the sequence of amino acids in those portions of the polypeptide chains are very similar between all antibodies. They don't vary much at all. So we call them the constant regions. Whereas at the top of the antibody, if I bracket this here, this is what's known as the variable region. So this portion here and this portion here, this varies between antibodies. So they'll have different sequences of amino acids in those variable regions. And this is important because at the top of the Y-shaped antibody, at the top of the variable regions, these are our antigen binding sites. Obviously, there are 2 of them. This is another one over here. So each antibody can bind to 2 antigens at the same time. And these have a specific shape that will be, we're going to use the word complementary to a specific foreign antigen. So the antigens can bind at those antigen binding sites. Be really careful here not to talk about active sites, okay? Yes, this is a protein. Yes, we've got the idea of complementary shapes fitting together, but it's not an enzyme. It does not have an active site. We should not be talking about it binding to a substrate. We should be referring to them as antigen binding sites binding to a complementary foreign antigen. Now, the fact that it has 2 antigen binding sites does mean that each antibody can bind to 2 antigens simultaneously, and it might bind to 2 antigens on 2 different pathogens, which will clump those pathogens together. And what we know about antibodies is that one of their roles is to cause agglutination, which is basically that clumping together of pathogens. So the fact that it has 2 antigen binding sites, it can bind to 2 antigens on 2 different pathogens, meaning it can agglutinate or clump together those pathogens, and then a phagocyte is able to come along and engulf and destroy multiple pathogens at one time. To help with this, this region here is called a hinge region. And it does allow the antibody to have some degree of flexibility, so it can maneuver itself and flex itself slightly to enable it to bind to 2 antigens on 2 different pathogens. I think I've covered everything. I hope you found this video useful. A lot of questions are about antibodies when it comes to the immune response, because ultimately we want to get to that end point, don't we, of plasma cells secreting antibodies so that phagocytes can engulf and destroy, or at least the antibody is marking the pathogen to attract phagocytes in order to destroy it.